Hi, my name is Joanna Claire Maressa. I'm a junior majoring in materials science and engineering. Today, I'll be talking about a three-dimensional tissue-engineered model of the blood-brain barrier during metastatic cancer. This research is conducted by the Searson Group within Johns Hopkins University Institute for Nanobiotechnology. I started working on this project in January of 2020. Since then, I've contributed to fabrication of the model, fluorescent imaging, and image and data analysis. Metastatic cancer is very common, and patients with breast cancer often experience cancer metastasis to the brain. This form of cancer is quickly lethal, so it's very important to develop treatments. Currently, no treatments are effective, and that's largely due to the blood-brain barrier, or BBB. The BBB is an organization of brain-specific endothelial cells, BMECs, pericytes, and astrocytes that line the capillaries in the brain. Its function is to regulate transport of molecules into the brain to keep anything out that could harm. However, that also means that it is significantly more challenging to treat medical conditions that occur in the brain. Many in vitro studies of brain cancers neglect the role of the BBB, which deems them less characteristic of the native human environment. In this project, my team's overarching goal is to develop a 3D tissue-engineered model of the BBB during metastatic breast cancer in order to better understand disease progression and to develop successful drug delivery strategies. I've been working on two objectives. The first is to characterize cancer cell spheroid growth to understand how we can quantify growth dynamics across various environmental conditions. The second objective is to study accumulation of an antibody treatment in cancer cell spheroids. If there are promising results, meaning there is significant antibody accumulation, that could promote the antibody in future studies as a potential anti-cancer therapy for metastatic breast cancer. Before we can run experiments, we need to make a model. On the left, you can see a clump of cancer cells. It's basically a tumor that we call a cancer spheroid. This spheroid is specifically of breast cancer that metastasizes to the brain. In the middle, you see a culture of derived human brain microvascular endothelial cells, or DHBMECs, that line the BBB. And on the right, you see a microfluidic device with a rod and inlet and outlet ports. This device is made with a PDMS scaffold, which is a polymer scaffold, and it's set onto glass. Once the device is fabricated, we have a mixture of cancer cells and collagen 1 and matrigel, which form a hydrogel that we insert into the device that acts as a tissue-engineered extracellular matrix. We then pull out the rod and we're left with a channel. In that channel, we can perfuse and seed these DHBMECs, which will then form the BBB metastatic model when the DHBMECs adhere to the sides of the channel. The inlet and outlet ports connect to reservoirs that allow for perfusion of nutrient-rich media through the microvessel, because normally through a vessel you'd have blood, but instead of blood, we're using the medium. When the device is fully fabricated, we can image it using fluorescent microscopy. Here is the fluorescent microscopy image of one device. In red, you can see the fluorescently tagged DHBMECs, the microvessel. In green, you can see fluorescently tagged spheroids. And the background in a blackish color is the collagen-1 matrigel hydrogel, which is the tissue-engineered extracellular matrix. Here's a confocal view of the DHBMECs, where you can see tight junctions that line the cells and show adhesion. Now that you've heard about the model setup, I can tell you about the experiments. Again, my first objective is to characterize cancer cell spheroid growth. In order to do so, I imaged the microfluidic devices with fluorescent microscopy on days 0, 2, 4, and 6. With these images, I analyzed them in a program called ImageJ. To analyze the spheroids, which is what we really want in these experiments, I put a perimeter around the spheroids. And within this perimeter selection, I could measure the area and how much the cells are fluorescing. In total, I analyzed 73 spheroids under flow, which I'll talk about later. In this specific device, you can see a time course of how this spheroid grows. And this area should be in microns squared, not millimeters squared. So we wanted to see how spheroids would grow under flow and static conditions. Under flow conditions, we perfused the nutrient-rich media through the BBB. Under static conditions, we didn't perfuse the media. We can measure which conditions led to higher spheroid growth by comparing the fold change in the area. Under flow conditions, there's significantly higher growth than under static conditions. That makes sense because the nutrients in the media can cross the BBB and reach the spheroids, thereby assisting in their growth. In this image, if you follow a spheroid on the left from day 0 to 6, you can see that it changes minimally, whereas on the right, there's an explosion in amount of green, which is spheroid cancer, that occurs over the 6 days. So here are some plots of the data that we can use to characterize the cancer cell spheroid growth. On the left, you can see the normalized spheroid area over time, which shows that cancer cell spheroids grow linearly proportional to time under flow conditions. On the right, you see normalized spheroid area versus their starting area, which means that when the spheroids are first put into the device, if they're smaller, they grow faster than if they're larger. We think that this occurs because there is a higher surface area to volume ratio in smaller spheroids, so it's easier for nutrients to reach larger proportions of the cells. In Objective 2, I evaluated accumulation of antibodies in the spheroids. I wanted to know if the antibody could permeate through the BBB and specifically accumulate in the cancer spheroids. The antibody we used is called Herceptin, and it's approved to treat metastatic breast cancer tumors overexpressing the HER2 receptor. However, it hasn't been approved for treating metastasis to the brain due to insufficient evidence. In terms of our experimental methods, Herceptin was perfused through the BBB to evaluate whether it could accumulate in the cancer cells. At the same time, I also perfused a non-specific IgG, meaning an antibody that doesn't specifically want to bind to the HER2 receptor. That way, we could tell if the Herceptin effectively targeted the HER2 receptor. Antibodies were perfused through the BBB on days 2, 4, and 6. You can see that the fluorescent intensity increased as time passed. To see where antibodies accumulated, I measured the mean gray value of the spheroids, the vessel, and the ECM. So the mean gray value I'm measuring within this ROI selection. The mean gray value measures the mean of the color inside the selection. This graph shows exciting results of this experiment. So In the extracellular matrix, there was barely any accumulation of either antibody, the Herceptin or the nonspecific IgG. In the vessel, there was some accumulation of Herceptin because the cells in the microvessel still have the HER2 receptor. It's just not overexpressed like it is in the cancer cells. And then when we analyze the spheroids, there is a larger increase in the Herceptin accumulation, which means that it successfully targeted the HER2 receptor. And the increase in accumulation of the nonspecific IgG did not occur as significantly. And now for the concluding summary of the presentation. So in Objective 1, cancer cell spheroids grow faster under flow conditions than static conditions. They grow linearly with time, and they grow faster when they are smaller initially. In Objective 2, we found that Herceptin can cross the BBB and specifically accumulate in spheroids due to the overexpression in the HER2 receptor. In the future, I'll be investigating whether spheroid-spheroid proximity and their spread across the extracellular matrix affect their growth. I'll also be investigating how the presence of cancer affects the BBB phenotype and how the presence of cancer affects gene expression in the BBB model using next-generation sequencing technology. Thank you so much for watching my presentation today. I'd like to acknowledge my mentors, Dr. Raleigh M. Linville, who mentored me heavily throughout this project, Alana Farrell, Dr. Jaubin Guo, and Professor Peter Searson. I'd also like to acknowledge the Johns Hopkins Institute for Nanobiotechnology and the Johns Hopkins University Department of Materials Science and Engineering. Here's a picture of all the fabulous people in my lab, and feel free to contact me at jmrs1@ghu.edu.